projects that really don't make a whole lot of sense. And sometimes these dollars, they go to projects having little or nothing to do with the public good. Things like fruit fly research in Paris, France. I kid you not.
Hold on there, Sarah. We have some researchers here who would beg to differ. Throughout history, Drosophila has been a useful model system to study genetics, and many of the signaling pathways present in flies are also present in humans. For the next few minutes, we would like to tell you about the world-renowned scientists who have won Nobel Prizes for their groundbreaking work studying Drosophila. We would first like to take you back to the turn of the 20th century and talk about Thomas Hunt Morgan, the scientist who made Drosophila famous.
Thomas Hunt Morgan was an evolutionary biologist, geneticist, embryologist, and author. In 1904, he established a lab at Columbia University, famously known as the Fly Room. He worked with colleagues Alfred Sturtevant, Calvin Bridges, Hermann Muller, and others on different aspects of fly genetics, including the chromosomal theory of heredity, genetic linkage, and crossing over. One of the earliest discoveries Morgan made with his flies proved that genes are on chromosomes. He crossed red-eyed males with white-eyed females and found that all daughters had red eyes and all the sons had white eyes. He called this crisscross inheritance because the sons had their mother's eyes and the daughters had their father's eyes. Morgan used cytology to link crisscross inheritance with the inheritance of the X and Y chromosome. He proved that the gene for eye color was on the X chromosome. This was the first proof that genes are on chromosomes. Morgan published a highly influential book called The Mechanism of Mendelian Heredity, which described genes located on chromosomes as beads on a string that contain important hereditary information. In 1933, he was awarded with the Nobel Prize in Physiology or Medicine for his discoveries concerning the role played by the chromosome in heredity. Morgan wasn't the only one from the Fly Room that won a Nobel Prize. One of his colleagues, Hermann J. Muller, used fruit flies to prove that heritable mutations could be induced by experiments done by humans. To do this, he used X-rays. He put flies in a box and then subjected them to massive doses of X-rays. This resulted in the formation of many mutations. With Morgan, he published 80 human-induced mutations in fruit flies, including yellow body color, miniature wings, and curly flies. This discovery was important for science because it made it possible to mutate genes rapidly to find out what their normal function is. The work was also important because it showed that humans can create changes in the environment that could lead to heritable changes in offspring. In 1946, Muller was awarded with the Nobel Prize in Physiology or Medicine for the discovery of the production of mutations by means of X-ray irradiation.
Almost 50 years later, the year is 1995. Edward Lewis, Christine Mussolini-Volhard, and Eric Weishaus win the Nobel Prize for their work with Drosophila. They worked on uncovering how embryonic development is controlled. While many of their colleagues thought this was an overly ambitious task, they went ahead and used clever genetics to discover all the genes required in the Drosophila embryo during early development. Their approach was to use saturation mutagenesis to test the effects of eliminating the function of every gene in the fly one by one. They discovered a total of 139 genes that, if mutated, caused defects on body axis formation and segmentation. The most interesting thing is, almost all have a correlate in humans. A particularly interesting group of evolutionarily conserved genes are the Hox genes. They found that the order in which these genes are orientated on the chromosome is the same as they are expressed in the body. This means that genes for the head region of the body will be on the opposite side of the chromosome for tail genes and abdomen genes in the middle. They also found several genes that were important in the patterning and formation of body segments, including gap genes, pair rule genes, and segment polarity genes. The gap genes are in control of creating and maintaining large regions of the embryo from head to tail. Pair rule genes affect every second segment during development. If a specific pair rule gene called even skipped is mutated, then the fly will develop with only the odd segments of their body fully formed. The segment polarity genes are responsible for creating polarity in each individual segment, ensuring the correct orientation of the segments in the fly. Their discoveries shifted developmental biology from a study of cells and metabolism to genes and genetic pathways. Significantly, they showed that fundamental mechanisms are conserved from flies to humans. Based on their pioneering work, the fly is now a major research organism used for understanding human disease.
16 years later, in 2011, Jules Hoffman, a Drosophila geneticist, shared the Nobel Prize with Bruce Beutler and Ralph Steinman for their work on immunity. Together, their data from fruit flies and mammals provides insight into how the innate and adaptive responses of our immune systems are regulated. Hoffman and his lab conducted research on flies to explore their antimicrobial mechanisms as a model for innate immune defenses in humans. The toll gene in flies was originally thought to solely be involved with embryonic development. However, Hoffman discovered that mutants without this gene were unable to mount defense against bacteria or fungi. This led to the conclusion that the toll gene was involved in the sensing of pathogens and that its activation is needed for defense. Toll-like receptors were then identified by Beutler that all recognize certain molecules common in microorganisms and activate innate immunity against them. These receptors have since been identified in humans as well. This data has the potential to be used to develop prevention and therapy techniques to fight against infections, cancer, and inflammatory diseases in humans. It is significant to note that Jules Hoffman is a French biologist who studies fruit flies and got a Nobel Prize for it. So, Sarah, after 4 Nobel Prizes, one specifically for fruit flies in Paris, France, what do you think about flies now?
Flies are cool!